Hi, this is Deb, and we're going to walk through some Punnett squares. I'm going to actually use the optional virtual labs that I put in there for you to use. You don't have to do these to turn them in, but I'd really encourage you to do them. It'll help you because there's questions about these on the test. And so if you've had this practice, you're in better shape. So let's go into Module 6, and you'll see that I put the one video I put up yesterday in there. And here at the bottom are optional Punnett square virtual labs. Let's start with the regular one. Laboratory exercise. All right, first off, let's watch the video.
This Punnett square predicts the results of a cross between two heterozygous tall pea plants. Each gamete carries only one of the two alleles that controls height in pea plants. The alleles from one parent are written over the top boxes of the Punnett square, and the alleles of the other parent are written to the left of the square. To determine the possible genotypes of all offspring, each box is filled in with the alleles to the top and the left side of that box. Each box then contains two alleles, one possible genotype. You can see that there are three different possible genotypes and yet only two possible phenotypes.
Okay, let's talk about those for a second. If you remember, this means homozygous, they're the same, and the capital T means it's dominant. So tall is dominant, and the probability is 1 in 4, every single time that dice is rolled and an offspring is formed, that you'll end up with that genotype of capital T, and the phenotype of a tall plant. And then 2 out of 4 times, you'll end up with these guys. Probability. So you have a heterozygous genotype, a big and a little, dominant recessive together, and then your phenotype, because that one dominant is there, your phenotype is going to be the dominant phenotype, what it looks like. So they're both going to be tall. So now we come to that 3-to-1 ratio that good old Mendel discovered. Then 1 out of 4 times, the probability is that you'll end up with a double recessive, a short plant. And this is the only way you can have a short plant, is if it's double recessive. Okay, now click out of that, and now you're ready for the experiments. All right, now we're in the lab. And every time you want to run a new scenario, they come up randomly, 1 through 10, you reset it. See? Okay, so let's start with scenario 1. Sounds pretty good. So we want to make a cross between a heterozygous gray-bodied. So let's look at our parent 1. It's going to be, heterozygous and gray, that one there in the middle. All right, with a homozygous black. And here we are. So now I've got my 2 guys, right? We better check it. Yay! And you'll see that it then took those gametes. So these are those chromosomes, those alleles out. So this guy had gotten a dominant and a recessive, maybe from mom a dominant, dad a recessive. And since those get divided during meiosis and only half of them go in, they're divided here. And then this guy, double recessive, so put both of those down, right? Now let's fill the squares up. So this is a big G and a little g. Yay! Little g, little g. Big G, little g. Little g, little g. Let's check offspring.
Fill in the rest of the Punnett square.
Oh my goodness.
The expected genotypes and phenotypes of all the offspring.
Oh my goodness, I just did the genotype, so I have to do the phenotypes. Yeah, sorry about that. So a capital G is going to be gray. All right, let's see if we can get it. Here's a double recessive, just like dad over here, for example. That's going to be black. Heterozygous, since you've got that one dominant, we're going to be gray. And double recessive, black. Now check them. Yay! Yay! All done. Okay, let's do another one. Make a cross between 2 flies that are expected to have all vestigial winged offspring. All right, vestigial, that's this little guy here. And Let's do our parents. And you'll see by looking at this that to have a vestigial wing, it has to be a double recessive. That's 2 lowercase l's, if I can pronounce it right. And if we went all recessive traits, that means our parents are going to have to be all recessive too. All right? So then we're pulling in, oops, oh my goodness, sorry, I have to check my parents first. Now, pull over. I'm going this guy with this guy, here and here, here and here, and the same for the last one. Now, let's put our guys in there. Since it's a double recessive, they all must have vestigial wings. Check offspring. Bingo. Okay, let's just do one more to make sure we've got this. And like I said, I really encourage you to go in and just do these until you really understand what's going on. It'll be great practice. And there are questions on the test about flies, exactly, actually. So you'll be doing these Punnett squares when you take the exam. All right, so scenario 5, make a cross between the following flies. Heterozygous long-winged with another heterozygous long-winged. All right, so parent 1, heterozygous, there's only one, that is one of each, and long wings dominant, so that's why they look like that. Now, let's check our parents. Yay, I actually remembered to do it. Okay, now let's start filling these in. So this is big L, big L. This one's little l, big L, so we'll do a big L and a little l. The larger one, the capital dominant trait, always goes first if it's heterozygous. Okay, big L, little l. Little l, little l. All right, now what are they going to look like? What's their phenotype? Because we just did their genotype. Long wings is dominant, the capital L, so that guy's definitely. And the heterozygous ones, because there's only one, even there's only one, it's dominant and it's going to mask that vestigial wing. So those guys are both going to have long wings. And then this guy, double recessive, only way we're going to get that recessive trait. Check offspring. Yay! All right. Okay, now we're going to take it a little bit different direction. We're going to look at X-linked Punnett squares. And to do that, we should do a little bit of background work. All right, first of all, this is a karyotype. This is actually a picture of all the chromosomes in a human's body. They're not lined up like this normally. we make them that way. So humans have 23 pairs of homologous chromosomes. And you can see them all. They're all different sizes, but they're together. And then the female, XX. That's what makes her a female. Or if it's a male, the 23rd is XY, right there. Now, so each one will only have a total of 46 chromosomes and 23 pairs. So either you got the female ones or you got the male ones. That makes sense, I'm sure. So what you want to look at here, and what makes a big difference, is how small that Y chromosome is right there. So what we're going to find out is that if there's a gene, a trait, an allele for that trait that happens to be right here on an X chromosome, there's no complementary part here to balance it out and make it be so you have to have 2 recessives to make it show. There's nothing against it. So if this has a recessive trait up here, this organism is going to have, this male is going to have that recessive trait. We'll talk more about it here in a second. So number 23 are those sex chromosomes, and 1 through 22 are called autosomal or body chromosomes. Every cell has all of these in them. So here's a better picture. This is actually a real picture. Here's an X, and they really do kind of look like an X. That's why they're called that. And there's the Y. And you'll see that a whole bunch is missing, it appears. It's not really, but it appears that way. So whatever traits happen to be on those chromosomes, on this Y down here, doesn't have anything here to balance it out. All right, so females have 2 X's, males X and Y. So let's say you have a female child. That means you got one of your X's, you are female, let's say. You got one of your X's from your mom, and then with that, it was a 50-50 chance whether you get this X or the Y in that sperm. It's only going to have one of them. And so if you're a female, you got your X from your dad. If you are a male, you got your X from your mom, and you got this Y from your dad. It's the only place you can get that Y. Your mom can't give it to you, only your dad. So males are XY, and the Y carries the SRY gene, which determines the maleness. So what makes the sex of your child, if you're having a child, is the male, is the sperm, is what's going to make it happen. So here's just a little bit more of that. All right. So the Y chromosome does not carry alleles for those traits. Some of those traits, like our male baldness and red-green color blindness, we're going to talk about that in a second. Those are some things that happen to be on that X chromosome and comes through. If a female has it on there but doesn't express it because she has another X that doesn't have that trait on it, so it's a recessive trait, so she's not going to have this showing, but she's a carrier because she can pass that X on to a son or a daughter. So, X-linked trait, red-green colorblind. So people with regular vision, we can see this. And if you're red-green colorblind, this is what it looks like. If you really are red-green colorblind, actually, the left side looks like the right side to you, I think. But you'll see this 45 over here doesn't show up, okay? So we talked about humans a bit. Let's move over to fruit flies for a second. So why do we use fruit flies? Man, they have huge chromosomes and only 8 instead of 23 pairs, right? So out of those 4 pairs, 1 are the sex chromosomes. And 2 through 4 are the body or autosomal chromosomes. And so here you see the picture. So once again, X is bigger, Y is smaller. Okay, we're going to go into more detail about this in a little bit, but just overview. I'm showing you the end result right off the bat. You see XX. These top 3 are females because they have XX, right? XY, right here, the 2 bottom ones, male, XY. So you see that really clearly. Then red-eyed happens to be dominant. So instead of having these letters by themselves on the X and Y on the X chromosome, they're a superscript. So a capital R means the same thing it means anywhere else that it's dominant. So red-eyed is dominant trait, and this female has both X's have the dominant trait of red-eye. All right. Then this one, dominant trait of red eye, recessive trait of white eye. So since she's got 1 red eye here, dominant, she's red-eyed. The only way she's going to be white-eyed is if she has 2 recessive traits. And it goes crazy here with this Y, little awry, so to speak. So the Y is not going to tell us anything, so whatever is on that X is what this male is going to be. So this capital R means this male is red-eyed, the dominant trait. Little r means he's the recessive trait. No ifs, ands, or buts. He's white-eyed. All right, so it kind of goes like this. This is sort of what you'll be building in just a few minutes. So here's the first cross, the F1 generation, and then they take this one. So here you have X with a capital R and X with a little r. Remember, you have to separate those out. And then here's Dad, X with a big R and a Y. And then fill it out, just like you did before. And now, we're just going to read it the same way, except for their superscripts. Same thing here. There's superscripts. And so we have a red-eyed female, but you see she's heterozygous. Red-eyed male, white-eyed male. Making sense? All right. Female, she's not, I'm colorblind. Now I switched to colorblind. I switched over to humans suddenly. It turns out that red-green I told you about in the beginning is on the X chromosome. So this female is homozygous dominant, so she's not colorblind. This female is heterozygous. She's not colorblind either because she has that one dominant allele right there. Then this one, homozygous recessive, just like before, colorblind. When you have the two recessives, even though it's on the X's, we got recessive. And just to make it easy, the last two are males, right? Because it's an X and a Y. And so whatever is on that first X chromosome, that's what that male is going to be. So not colorblind and colorblind. And here's just a little thing showing, like if your grandfather was colorblind, let's trace this X with a colorblind gene that he passed down to his daughter. Here it is right here. And she passed it down to this daughter, but it didn't have any effect because this daughter got that dominant trait for normal vision from her dad. But this one was a boy, so as this one came right through here, boom, colorblind son. Okay, I've gone back into our class and opened up the link to the sex-linked traits and opened up this virtual lab. This is more we explore and see what we've got. Let's see. So you open up the lab book like we did before. I've already saved us a little time. All right, let's say the male and let's have female. All right. So, he should have red eyes up here, by the way. That's irritating. All right. So, now we pull these down just like we did before. This will be an XX, so this will be a female, and they'll both be the dominant trait. So do we find that one? Yay, right there. So we put this guy in. Now let's go over to, so we know this is a male in this spot, the male with red eyes, because it has a dominant R, capital R there. Here we have big R, and big RY. And we got all red eyes. And remember, this guy's wrong. This guy up here, the parent is supposed to have red eyes. I can't believe that they made that mistake. So let's check. Yay! All right, let's do another one. Okie doke. Let's see. Let's have really a wide-eyed male with a heterozygous female. Okay, heterozygous, remember, different. All right, so now, boom, makes sense? And then, there you go, big R, Y, and then, oh, my goodness, I didn't even plan that. I just created a wide-eyed female. That's kind of rare, actually. It's rarer than the other ones. And a wide-eyed male. Let's check. All right. So hopefully you have an idea how to do these. I'd encourage you to practice them a bit. These virtual labs are in there, so you can use them. And next, we'll go on to what if we want to cross two traits.